Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, materials science, chemistry, and physics, all told as compelling, easy-to-follow stories. And today we are talking about a word that you have probably seen on, everything, from fancy pomegranate juices at the grocery store to those ridiculously expensive anti-aging face creams. I mean, I'm talking, of course, about antioxidants.
Yeah, they really are everywhere. I mean, you literally can't walk through a pharmacy or a supermarket without having that word aggressively marketed to you. It's become this modern health halo.
Right, we basically treat them like microscopic superheroes. They're sold to us as the Avengers of the cellular world. And the marketing narrative is so simple, right? Their sole job in your body is to just punch the quote-unquote evil free radicals in the face to keep you young and vibrant.
Exactly. Disease-free and glowing. That's the pitch.
But here's a really strange question for you. If antioxidants are the ultimate biological good guys, why did massive scientific trials involving high-dose antioxidant supplements completely fail? And like some of the most famous cases, why did they actually make people sicker?
It is arguably one of the most fascinating paradoxes in modern human biology. Scientists did eventually unravel this mystery, but the answer requires us to completely tear down our fundamental understanding of health and, well, cellular stress.
Oh, wow, really?
Yeah, absolutely. So to figure out what actually happened, we are diving into a pretty hefty stack of foundational and cutting-edge literature today. We are drawing heavily on Dr. Barry Halliwell's foundational work, including his comprehensive 2024 review on antioxidant mechanisms.
Okay.
We also have G. Pizzino's 2017 review on oxidative stress, which was published in the journal Oxidative Medicine and Cellular Longevity, plus a critical 2022 review from the journal MDPI by Kotha and colleagues regarding antioxidant assays.
That is a lot of ground to cover.
There's a bit more, actually. We are also looking at I. Gulson's 2025 comprehensive review, and finally, a Web of Science bibliometric landscape analysis by Andy Y. Kanyung.
Awesome. So basically, our mission for this deep dive is to look at all of this data to figure out what antioxidants actually do inside your body. We want to understand the specific molecular mechanics of why the old rule of, more is always better turned out to be a total myth.
Right, that's the goal.
So to start us off, let's go back to the original scientific consensus. We used to view our bodies sort of like a car rusting in the rain, right?
That is a very accurate representation of the old model, actually. For decades, the view in biochemistry was, well, it was purely black and white. As your cells generate energy, they continuously produce these unstable molecules known as free radicals as a natural byproduct.
Right, the exhaust from the engine, basically.
Exactly. We are specifically talking about reactive oxygen and nitrogen species, which are usually abbreviated as ROS and RNS. Things like superoxide and hydrogen peroxide are the classic examples. And because these molecules are missing an electron, they are highly reactive. They basically run around your cells trying to steal electrons from literally anything they bump into.
And when they steal that electron, they cause oxidative damage. So the logic was that these ROS were just, tearing up our DNA, our proteins, and our lipids.
Yep.
And that was leading directly to aging, cancer, and neurodegeneration.
Yeah, that was the thought process.
So we assumed the car was rusting, so we needed a rust-proofing spray. And we thought antioxidants like vitamin C and vitamin E were that spray.
That was the exact assumption. And, to be fair to the early researchers, it made perfect sense based on their preliminary observations. In early lab experiments, if you exposed a cell culture to high levels of free radicals, the cells sustained massive damage.
Makes sense.
Right. But if you flooded the culture with vitamin C, the damage halted. So the medical community and the supplement industry made this massive leap of faith.
Uh-oh. Let me guess. They rushed to market.
Basically, yeah. They figured, let's just give people massive doses of isolated antioxidants in pill form, eradicate oxidative stress entirely, and we'll wipe out heart disease and cancer in a generation.
Wow. But biology is rarely that straightforward, is it?
Wow.
So how did scientists figure this out? When researchers finally ran these massive randomized controlled trials, giving well-nourished people high-dose isolated supplements, the results were a colossal shock. I mean, we aren't just talking about a slight statistical miss.
Oh, from it. Large-scale supplementation studies using isolated vitamin E and vitamin C routinely showed zero significant reduction in cardiovascular events or cancer incidents. Nothing.
Nothing at all.
Nothing. But the real wake-up call was a specific trial involving beta-carotene, which is it's the antioxidant compound that gives carrots their orange color.
Okay, yeah, I've heard of that one.
So researchers set up this massive trial giving high-dose beta-carotene to heavy smokers. They assumed it would protect their lung tissue from the intense oxidative stress caused by cigarette smoke.
Right, on paper, that sounds perfectly logical. You take a highly toxic, oxidizing environment like a smoker's lung, and you flood it with the ultimate biological rust-proofing spray.
You would think so, yeah?
But the trial actually had to be stopped early, didn't it?
It did, and, well, the results were the polar opposite of what anyone expected. The high-dose beta-carotene supplements didn't protect the smokers. They significantly increased the participants' risk of developing lung cancer.
Wait, increased it.
Yes, it was a spectacular backfire.
This is where the story gets really interesting. Because if water puts out a fire, why did bringing a fire hose to this cellular fire make the house burn down faster? Like, how do you go from this molecule cures aging to this molecule actively fuels cancer?
To answer that, the scientific community had to completely reevaluate what free radicals actually do in the human body. And this brings us to a crucial turning point in the research, which is heavily detailed in B. Halliwell's 2024 review and Pizzino's 2017 findings.
Okay, what do they find?
Scientists realized they had profoundly misunderstood reactive oxygen and nitrogen species. They aren't just destructive rogue molecules. At controlled, low levels, they are absolutely essential signaling messengers.
Okay, I want to pause on that word messengers. Are you saying the free radicals themselves are communicating information inside the cell? Because I always pictured them as just mindless demolition crews.
No, it is actually a highly sophisticated communication network. Your body intentionally produces these free radicals to send signals.
Intentionally?
Yes, intentionally. Here's how it works mechanically. Proteins in your cells have these specific amino acids, like cysteine, which act almost like biological switches.
Yeah, I'm following.
When a free radical like hydrogen peroxide interacts with that cysteine, it oxidizes it. That oxidation slightly changes the shape of the protein, which turns a specific cellular pathway on or off.
So the free radical is basically acting as a molecular key, turning on systems that regulate, like cell division, immune responses, and survival pathways.
You've got it. Your immune system even uses bursts of free radicals as chemical weapons to destroy invading pathogens. Your cells use them to talk to each other and to adapt to changes in your environment. So complete suppression of ROS is not just impossible, it would be instantly fatal.
Wow. So by taking massive antioxidant pills to eliminate all free radicals, we were essentially cutting our own biological phone lines. We silence the very alarm bells our bodies use to detect problems and adapt to them.
That is a brilliant way to conceptualize it, yeah. You are blinding your cells to their environment. But as wild as that is, it only solves half the mystery.
Right, because of the smokers.
Exactly. The silenced alarm bell theory explains why the antioxidant pills might not prevent disease, but it doesn't explain why the beta-carotene actively caused harm in the smoker trial. To understand that, we have to look at the chemistry.
Let's get into it. Why did the supposed firefighter suddenly become an arsonist? If I take a massive dose of vitamin C, its entire molecular purpose is to donate an electron and stabilize a free radical. How does giving away an electron cause a fire?
It comes down to what it gives that electron to. This happens through a process called Fenton chemistry.
Fenton chemistry, okay.
Yeah. Under normal conditions, an antioxidant like ascorbates, vitamin C donates an electron to a radical to neutralize it. But if you dump a massive unnatural concentration of ascorbate into your system via a supplement, it starts interacting with transition metals floating around in your body, specifically free iron or copper.
Okay, so the vitamin C bumps into iron. What happens then?
The vitamin C donates its electron to the iron, reducing it. That newly modified iron is now highly reactive, and it immediately reacts with relatively harmless peroxides that are naturally present in your body.
And what does that reaction do?
It transforms those mild peroxides into something called hydroxyl radicals. And hydroxyl radicals are the most dangerous, destructive free radicals known to biology.
Wait, really? Why are they so much worse than the normal free radicals our mitochondria make?
Because of their reactivity rate. Hydroxyl radicals operate at what chemists call the diffusion limit.
Diffusion limit, what does that mean?
That means they instantly destroy the very first molecule they bump into, whether that's a piece of DNA, a lipid membrane, or a critical protein. You can't even scavenge them once they are created. They react too fast.
It's like hiring a massive muscular bodyguard to protect your house, but you pay him so much that he gets completely bored, gets drunk, and starts smashing your own furniture to pieces.
That is a perfect analogy. And this pro-oxidant behavior is highly context dependent. It depends heavily on the dose, the specific oxygen environment of your tissues, and the availability of those metals.
So back to the smoker study.
Right. If we look back at that study with the smokers, you took a massive dose of isolated beta-carotene, dropped it into the highly oxygenated, highly toxic environment of a smoker's lung, which is already full of unstable molecules and disrupted metals from the smoke, and you essentially provided the raw fuel for a massive Fenton reaction.
Oh, wow.
Yeah, you just created a cascade of pro-oxidant damage that directly damaged their DNA and accelerated the cancer.
But if all this is true, I have to ask, how did we get it so monumentally wrong in the first place? I mean, I read the MDPI critical review by Kotha and colleagues. It seems like the early lab tests completely misled us.
They absolutely did. According to I. Goldson's comprehensive 2025 review and the Kotha paper you mentioned, the fundamental flaw was relying on in vitro assays. Those are tests done in a glass tube or a plastic dish outside of a living organism.
Right, the classic test tube trap. But what exactly is the trap here? If a chemical neutralizes a free radical in a glass tube, why wouldn't it do the exact same thing in my liver?
Think about the complexity of your liver versus a glass tube. One of the most famous early tests was the DPPH assay. You basically take a purple liquid containing a stable synthetic free radical.
Okay.
You drop in your antioxidant extract, maybe from a blueberry or a new supplement. If the chemical donates an electron, the purple liquid turns clear. Researchers would see that and declare, aha, high antioxidant activity.
But a test tube full of purple liquid doesn't have transition metals floating around waiting to trigger a Fenton reaction.
Exactly. I mean, you are right on the money. A test tube doesn't have dynamic oxygen gradients. It doesn't have a liver metabolizing the compound into different byproducts.
Right.
It doesn't have iron bound up in ferritin that gets released during cellular stress. A chemical might happily turn that liquid clear in a lab, but act as a violently destructive pro-oxidant inside the complex in vivo environment of a human cell.
So how do we actually measure it today if the purple liquid is useless?
The literature now stresses that to understand what happens in humans, we have to look at actual biomarkers of damage inside the body rather than theoretical scavenging capacity. A prime example is a biomarker called 8-oxo-DG.
Break that down for us. What is 8-oxo-DG?
It stands for 8-oxo-2-deoxyguanosine. Guanosine is one of the 4 main bases that make up your DNA.
Okay, the building blocks.
Right. When a severe free radical, like the hydroxyl radical we talked about, attacks your DNA, it oxidizes that guanosine base. If the cell divides without fixing it, that oxidized base causes a mutation, which can lead to cancer.
Ah, I see.
So by measuring the levels of 8-oxo-DG in a person's urine or blood, we aren't just guessing if an antioxidant works based on a color change in a tube. We are directly measuring whether DNA damage in the human body is going up or down.
And relying on the tube instead of the human body is what led to those failed clinical trials. Which brings me to a really interesting realization. This perfectly explains why eating a handful of blueberries is demonstrably good for you, but taking a megadose blueberry extract pill might do absolutely nothing, or even cause harm.
You're hitting on the exact current scientific consensus. The evidence, supported by decades of epidemiological studies, strongly favors dietary patterns over isolated pills.
Right.
When you eat a whole food, fruits, vegetables, whole grains, legumes, you aren't just getting a massive dump of a single isolated vitamin, you're getting a complex synergistic matrix of bioactives.
But wait, let me push back on that a bit. When you say complex matrix, it sounds a bit like marketing jargon.
Oh, yeah.
What is actually happening biologically when I eat the blueberry that doesn't happen with the pill?
It all comes down to a concept called hormesis, the biological principle that a small amount of stress makes a system stronger. This is where the story gets really wild.
Okay, I'm ready.
The phytochemicals in that blueberry, the compounds that give it its color and flavor, are actually plant defense chemicals. To the human body, they are very mild toxins, or xenobiotics.
So you're saying the healthy chemicals in vegetables are actually mildly stressing my body out?
Yes. When you eat them, they cause a tiny, manageable spike in cellular stress. Your cells detect this mild threat and respond by activating a master genetic switch called the NRF2 pathway.
Ah, I read about NRF2 in the Halliwell review. Let me see if I have the mechanics right. Under normal conditions, NRF2 is anchored outside the nucleus of the cell by a sensor protein called KEAP1.
Right.
But when the cell experiences a tiny bit of oxidative stress-like from those plant chemicals, KEAP1 changes shape and lets go. NRF2 then travels into the nucleus and turns on the body's own internal antioxidant factories. Did I get that right?
That is a phenomenal summary, yeah. It travels to the DNA and tells your body to manufacture its own endogenous or internal antioxidants. We're talking about incredibly powerful enzymes like superoxide dismutase, catalase, and glutathione.
And they work differently than the pills.
Oh, completely. These endogenous enzymes don't just neutralize one free radical and die like a vitamin C molecule. They can neutralize millions of free radicals per second, and they do it safely without triggering Fenton chemistry.
So the food isn't just delivering antioxidants, it is acting as a biochemical training camp, forcing my own cells to build up their internal defenses.
That is the crucial paradigm shift. And it explains why healthy lifestyle factors like exercise work the exact same way. When you go for a run, your muscles consume a massive amount of oxygen and your mitochondria generate a huge wave of free radicals.
Because you are intentionally causing oxidative stress.
Exactly. But that pulse of ROS triggers the exact same NRF2 pathway, beefing up your internal defenses.
This means we need to stop treating our biology like a simple math equation where, stress minus antioxidant equals health. Your body isn't a math problem. It's a highly dynamic ecosystem.
Precisely.
It actually needs a little bit of stress, a little bit of damage to trigger its own defense systems and stay resilient. If you constantly swallow mega doses of antioxidant pills, you're sheltering your body so much that its own internal defense systems get weak and lazy.
And you are seeing this shift happen in the scientific community right now. If we look at A.W.K. Young's bibliometric landscape analysis from the Web of Science, we can actually track this evolution in real time.
What are they finding?
The research funding and focus is moving rapidly away from isolated vitamins and towards what are called targeted cytoprotective agents.
Cytoprotective agents, meaning they protect the cell, but not by just acting as a simple free radical sponge.
Correct. Scientists are highly focused now on unique molecules like ergothioneine and reactive sulfur species. Ergothioneine, for example, is a unique amino acid found heavily in mushrooms.
Okay, what makes it so special?
What makes it special is that the human body actually evolved a highly specific transporter protein just to pull it into our cells and concentrate it in tissues that undergo high oxidative stress.
Oh, wow.
Yeah, it doesn't just indiscriminately quench radicals. It helps tune those complex redox signaling networks we talked about earlier. It acts as a modulator, not an eraser.
That is fascinating. We evolved a specific cellular doorway just for this one molecule. Are there other applications for this new understanding outside of just diet and supplements? Because the Young analysis mentioned we're moving into some pretty futuristic territory.
Oh, absolutely. One of the most exciting future frontiers is the development of antioxidant biomaterials. This represents a massive leap in how we treat severe, localized oxidative stress.
Localized stress, meaning what?
Well, instead of swallowing pills to flood your whole systemic circulation, scientists in fields like tissue engineering are developing synthetic scaffolds and natural polymers that have antioxidant properties built right into their molecular structure.
So instead of systemic suppression, we're talking about spatial control. We physically place a material into the body.
Precisely. It is all about location. Imagine a patient with a severe diabetic ulcer, a chronic wound that simply will not heal. In a wound like that, the local immune cells like macrophages get stuck in a hyperinflammatory loop.
Right.
They are continuously spewing out massive amounts of destructive ROS, which tears down new tissue as fast as the body can build it.
And if you gave that patient a massive systemic antioxidant pill, it might not even reach the wound in high enough concentrations, but it would mess up the healthy signaling pathways in their brain or liver.
You nailed the problem. So the new approach is to apply an antioxidant polymer like a specialized hydrogel directly into the wound bed.
Oh, I see.
The material's molecular structure is designed to scavenge the harmful ROS only in that specific location. It brings the local fire down to a manageable level so the tissue can finally regenerate, but it leaves the crucial ROS signaling pathways in the rest of the body completely untouched. It is highly targeted spatial therapy.
That is a staggering leap from the old days. It really highlights how far the science has come since the era of just staring at a purple liquid turning clear in a test tube and assuming we'd cured aging.
The evolution of the science is truly remarkable. So to synthesize this entire journey for you today, there are really three major takeaways from the current scientific consensus.
Let's hear them.
Number one, Free radicals are not just cellular villains. At controlled levels, they are vital communication molecules that your body needs to survive and adapt. Number 2, megadosing isolated antioxidants can completely backfire. It can silence your biological alarm systems and even cause severe pro-oxidant damage via Fenton chemistry.
The arsonist firefighter.
Exactly. And number 3, the most robust, scientifically proven way to protect yourself is not through a magic pill, but through complex dietary patterns and lifestyle habits like exercise that gently stress your body and force it to tune up its natural endogenous defense networks.
It is a profound shift in perspective. Science is rarely black and white, and this story completely proves it. The very molecules we thought were purely destructive are actually essential for life. And the supposed quote-unquote cure can become deeply toxic if taken completely out of context.
Perfectly said.
Which leaves you with something to really think about. If our bodies actually rely on a precise, delicate balance of damage and stress to stay resilient and healthy, well, what other quote-unquote bad things in our biology might actually be the secret to our survival?
That is a brilliant question. And, it really challenges everything we think we know about what it means to be healthy.
It sure does. Well, we've unpacked a lot of complex chemistry and biology today. Thank you so much for joining us on this investigation in the Deep Dive Lab. If you enjoyed this journey into the microscopic mysteries of your own body, be sure to follow and subscribe for more science stories. See you next time.